Namaskar. Welcome to our channel Bio by Gulab Sir by Wisdom Coaching Center. As a series of lecture, today we are going to learn about biochemical characterization of transforming principle. We already had discussion on transforming principle given by Frederick Griffith. Now, in continuation of that experiment, as a series of experiments to prove that DNA is the genetic material and not protein. So we are continue to have explanation for Griffith experiment given by MacLeod, McCarty and Avery. So heading is characterization or biochemical characterization of biochemical characterization of transforming principle given by three scientists. The name of scientist is Oswald, Avery, Colin, MacLeod and McCarty. These three scientists, they gave a biochemical explanation for Griffith transforming principle. What they done? They made an extract of that S-shaped bacteria. Extract from S-shaped bacteria. What they have extracted? Because they were supposed to prove that DNA is the genetic material and not protein and RNA or not other principle present inside the 8-kill bacteria. So they made extract from S-shaped bacteria. What that extract? One, as a protein, then RNA and DNA. Now one by one they injected into the mice along with R-shaped bacteria. They injected these into the mice along with R-shaped bacteria. So injected with R-shaped bacteria into mice. So when they injected this into mice, that in along with R-shape in mice, here in these two cases, first two cases, mice survived. Here mice died, and they also do discover, they have discovered enzymes such as proteases, RNase and DNase. And when they use these proteases along with this, there was no effect. But when they use DNase along with these, there was effect. What was that effect? That mice did not die. Why? That DNase could digest this DNA and therefore could not convert R-shape into S-shape, and thereby mice survived, because R-shape do not cause disease, and since it did not convert into S-shape, that means there was no disease and died. That is what explanation given for it is like this. They injected DNA with R-shaped bacteria plus DNase enzyme, that nuclease enzyme. Now since DNase digests the DNA, therefore R-shape will not convert into R-shape. So injected into the mice and injected into mice. So here it is found that mice survived. Means because of DNA digested, Since DNA is digested, there is no DNA in that combination, therefore R shape is not converted into S shape to cause disease to the mice and died. Therefore, R shape remain R shape, therefore mice survive. It means that control over experiment, that when they use proteases, there was no any change. They use DNase, RNase, there was no change. But when they use DNase along with this DNA and R shape, that there was a change, because along with DNA, when injected into the mice along with R shape, mice was died. It means DNA could have converted R shape into S shape and mice got pneumonia, and from that dead body they could extract S-shaped bacteria, and it is found that DNA would have transferred R shape into S shape to cause pneumonia and died. But here they did not got S-shaped bacteria in mice. That confirms DNA is the transforming principle of Griffith experiment, because DNA transferred R shape into S shape. It is because of that control over experiment done by MacLeod, McCarty and Avery by using different enzyme, proteases, RNase and DNase. When they add DNA, R shape and proteases, there was no change. That R shape remain R shape. When they use DNA, RNA, R shape and proteases, that mice got died. When they use DNA, R shape and RNase, mice got died. But when they use DNA, RNase and DNase, then mice survived. That means DNA would be the genetic material to convert R shape into S shape and cause pneumonia to the mice. So I hope it is very clear to you how that MacLeod, McCarty and Avery has given biochemical characterization for transforming principle by extracting these three components. Out of the three components, these two components did not convert R shape into S shape, and therefore no disease to the mice got survived. But when DNA of S-shaped bacteria injected along with R shape into the mice got died. To have the control over experiment to prove that why DNA only but not RNA and protein, that they have discovered three enzymes, proteases, RNase and DNase. When they use these three enzymes along with these DNase with R shape, proteases, that mice died. DNA with R shape, RNase, mice got died. But when DNA with R shape and DNase, mice got survived. There was a change when DNase was used. That shows that DNase must be genetic material. And this is the experiment given by these three scientists. Question can be asked in board, that explain biochemical characterization from transforming principle given by MacLeod, McCarty and Avery for 3 marks. Or explain experiment given by MacLeod, McCarty and Avery to prove that transforming principle given by Frederick. Or what is transforming principle? Explain an experiment given by MacLeod, McCarty and Avery to prove that transforming principle is a DNA and not the protein. So this question can be asked, and I am sure that if any question asked in NEET, you will be in position to answer the question based on application of knowledge and understanding to the question, as concept is clear to you. Once again, to clarify that when they did use these three enzymes, that these three enzymes are proteases, RNase and DNase along with this combination. When they use this DNA R-shaped bacteria and proteases, there was no change, that mice died. When they use DNA R-shape and RNase, mice died. But when they use DNA R-shape and DNase, mice survived. It is very clear to you. No need to write that. With the use of protease and RNase, there was no any change. Its statement is given in book, that this combination, when proteases and RNase used, there was no change. But when DNA is used, mice got survived. This shows that DNA is the genetic material. I am sure that you would have understand and you like this. If you are in doubt, you can ask in comment section. I appeal you to like our channel by subscribing it and by pressing bell icon you will be notified when new videos will be uploaded. Thank you.